Acha bete. So in today's topic, learning objectives are explain the relative energy values of carbohydrate, lipid, and protein as respiratory substrates, and explain why lipids are particularly energy rich. So बेटा आपके ऐसे टाइप क्वेश्चन में ना ये respiratory substrates का एक 6 marks का क्वेश्चन अक्सर आ जाता है. ठीक है, we'll discuss that. Then define the term respiratory quotient and determine respiratory quotient from equations for respiration. So this is what we are going to do also. We are going to discuss these also today. Explain the production of a small yield of ATP from respiration in anaerobic conditions in yeast and in mammalian muscle tissue. As well as we are going to explain how rice is adapted to grow in its roots submerged in water in terms of tolerance to ethanol from respiration in anaerobic conditions, and carry out investigations using simple respirometers to measure the effect of temperature on the respiration rate of germinating seeds of small invertebrates. So for this bit, I'll show you a video, okay, and then we'll see how we can relate that to your P5 questions then. Okay, so let's start with the lesson then. Okay, bete. So okay, previous lesson में हमने क्या discuss किया था? All about the aerobic respiration. Okay, and we were getting like a lot of ATPs there. Okay, at the end, oxidative phosphorylation के बाद. But sometimes beta humans will also revert to anaerobic respiration. ठीक है, और generally that would happen when let's say they are exercising. ठीक है, तो oxygen जब concentration कम हो जाएगी तो they would revert to anaerobic respiration. It means there is no oxygen involved at all. So generally beta when we talk about organisms जिनका primary ही method of respiration anaerobic है, those are known as anaerobes. ठीक है, the process beta wherever oxygen is not used for respiration is known as fermentation. ठीक है, अच्छा, but we will be talking about the process both in humans and yeast. So because these are the two examples that are given in a book also. ठीक है, so दोनों cases में beta if you recall glycolysis is happening in the cytoplasm and pyruvate is being formed. Because glycolysis वाले case में क्या हो रहा था? Only glucose was being broken down. Glucose was a substrate. It was being broken down into pyruvate. Remember that beta? ये चीज रिकॉल हो रही है बेटे? Glucose is a 6 carbon compound, ठीक है, और glycolysis में it was converted into 2 molecules of 3 carbon compound which is pyruvate. ये चीज याद है बेटे? हमने लास्ट लेसन में डिस्कस किया था. Yes, no?
Yes, no.
अच्छा, so glycolysis तो aerobic और anaerobic दोनों के अंदर exactly same है. ठीक है, उसके बाद के प्रोसेसेस चेंज हो रहे हैं. ठीक है, अब व्हाट हैपेंस बेटे, इट स्टार्ट्स विद ग्लूकोज बट एट द एंड दोनों के जो एंड प्रोडक्ट्स होंगे, बिकॉज वी विल बी टॉकिंग अबाउट बेटा कि ह्यूमंस में उसका एनारोबिक रेस्पिरेशन से क्या प्रोडक्ट बन रहे हैं और एनारोब्स में ईस्ट के अंदर क्या प्रोडक्ट बन रहे हैं. सो बोथ ऑफ द प्रोडक्ट्स बेटा व्हेन दे फॉर्म आर टॉक्सिक इन लार्ज अमाउंट्स एंड दे हैव टू रिमूव फ्रॉम द सेल्स आल्सो. ओके, सो वी वर डिस्कसिंग बेटा कि इन एनारोबिक रेस्पिरेशन, ठीक है, we will get ATP but we will only get a small yield of ATP. ठीक है, तो ऐसा नहीं है कि नहीं मिलेगी. So whenever there is a requirement, body shifts towards anaerobic respiration. ठीक है, so it is required whenever we get short rapid burst of ATP. So this usually better happens during exercise. ठीक है, anaerobic respiration will also take place when respiring cells run out of oxygen. ठीक है, और प्लांट्स के अंदर कहाँ पर होगी? सो बिकॉज समटाइम्स देयर इज एन एनवायरनमेंट दैट इज डेफिशिएंट इन ऑक्सीजन, सो वाटर लॉग सॉइल्स के अंदर भी प्लांट्स विल रिवर्ट टुवर्ड्स एनारोबिक रेस्पिरेशन. ठीक है, सो एज पर नीड प्लांट्स या एनिमल्स कैन रिवर्ट टू दैट, बट उसके अलावा कुछ एनारोबिक ऑर्गेनिजम्स होते हैं, फॉर एग्जांपल यीस्ट, तो दे विल एक्चुअली बेटा रिवर्ट टुवर्ड्स दिस. अच्छा, अब इस पूरे प्रोसेस में बेटा द थिंग इज फ्री ऑक्सीजन इज नॉट प्रेजेंट. And if you recall from aerobic respiration, बेटा वहाँ पे ऑक्सीजन का एक बहुत बड़ा रोल था ऑक्सीडेटिव फॉस्फोराइलेशन में. So can you recall बेटा what was happening?
Ma'am, it was the final electron acceptor.
Good. So अब अगर ऑक्सीजन मौजूद ही नहीं है बेटे, so it will not be accepting those electrons. ठीक है, और remember बेटा क्योंकि ग्लाइकोलिसिस में if you remember आपके पास 2 reduced NADH बनते हैं. ठीक है, अब ये वाली हाइड्रोजन जो बेटा it has to be dumped in any way. तो अब जब इलेक्ट्रॉन एक्सेप्टर ही हमारे पास फाइनल स्टेज में नहीं है, तो फिर ये हाइड्रोजन क्योंकि बना तो है ग्लाइकोलिसिस में, इसको रिमूव कैसे किया जाए? ठीक है, सो फॉर दिस प्रोसेस बेटा एनारोबिक रेस्पिरेशन में सी बेटा हाउ दिस हाइड्रोजन इज रिमूव्ड, राइट? तो वी वर जस्ट सेइंग बेटा कि इलेक्ट्रॉन ट्रांसपोर्ट चेन विल नॉट हैपन. तुमने मुझे खुद ही बताया ऑक्सीजन इज द इलेक्ट्रॉन एक्सेप्टर. ठीक है, सो दिस इलेक्ट्रॉन ट्रांसफर चेन बेटा यही वाला जो इवेंट होगा, दिस वुड स्टॉप वर्किंग. इफ इट इज नॉट वर्किंग एट ऑल, नो फर्दर ATP is formed. और क्योंकि एटीपी सबसे ज्यादा मेजर अमाउंट ऑक्सीडेटिव फॉस्फोराइलेशन के अंदर ही बन रहा है, ठीक है। बट ग्लाइकोलिसिस में भी रिमेंबर बेटा 2 एटीपी मॉलिक्यूल्स बनते हैं, ठीक है। इनफैक्ट लेट मी शो यू द डायग्राम फर्स्ट, तो एक हम इसको वहां से रिलेट कर लेते हैं, ठीक है। बिकॉज बेटा इवन इन ग्लाइकोलिसिस वी आर गेटिंग दिस 2 एटीपीज, ठीक है। आपको एटीपी, आपको फाइनल प्रोडक्ट जो था 2 एटीपीज हमें मिल रहे हैं वहां पे, सही है। बट एटीपी के साथ साथ यू आर गेटिंग टू रिड्यूस एनएडीएच हमें, क्योंकि एक ग्लूकोज का मॉलिक्यूल बेटा 2 मॉलिक्यूल्स ऑफ पायरुवेट में कन्वर्ट हुआ था, तो फ्रॉम ईच मॉलिक्यूल ऑफ पायरुवेट वी आर गेटिंग दिस रिड्यूस एनएडीएच, ठीक है। एंड दिस हाइड्रोजन हैज टू बी रिमूव्ड आल्सो, ठीक है। एंड यू आर गेटिंग एटीपी एज वेल, इवन इन लोअर अमाउंट, बट हमें एटीपी मिल रही है, ओके देन, ठीक है। सो दिस एटीपी मॉलिक्यूल्स आर actually formed there in glycolysis, but this hydrogen has to be better passed on, okay. So how this hydrogen will be removed from it? So the 2 anaerobic pathways that we will talk about is बेटे एक हम बात करेंगे formation of ethanol fermentation की, ठीक है, और दूसरी हम बात करेंगे lactic fermentation की, ठीक है। So the basic aim of this anaerobic respiration is to dump this hydrogen. This hydrogen has to be removed. और रिमेंबर जितनी भी एनाएरोबिक रेस्पिरेशन की हम बात करेंगे, एवरीथिंग विल बी टेकिंग प्लेस इन द साइटोप्लाज्म ऑफ द सेल। अब माइटोकॉन्ड्रिया का कोई रोल है ही नहीं यहाँ पे, ठीक है। अच्छा एक मिनट देना बेटा मुझे। अच्छा बेटे, सो नाउ वी विल बी यूजिंग द टर्म फर्मेंटेशन, ठीक है। अब फर्मेंटेशन सिंपली क्या बेटा, कैटाबॉलिक प्रोसेस। कैटाबॉलिक क्यों कह रहे हैं, बिकॉज ब्रेक डाउन इज टेकिंग प्लेस, ठीक है। सो कैटाबॉलिक प्रोसेस दैट मेक्स अ लिमिटेड अमाउंट ऑफ एटीपी from glucose without an electron transport chain. So this is the basic definition of fermentation बेटे, ठीक है। And it produces end product. तो अब अगर बेटे हम लोग वहां पे anaerobic respiration by yeast की बात कर रहे हो, तो your end product will be ethyl alcohol या ethanol, ठीक है। या अगर हम बात कर रहे हैं anaerobic respiration taking place by mammals, ठीक है, जो डेफिनेटली बेटा in case of exercise अगर for example हो रही है, तो that will be known as lactic acid, ठीक है। बोथ ऑफ देम आर फर्मेंटेशन, ठीक है, बट सिंपली एंड प्रोडक्ट्स डिफरेंट है। ईस्ट के अंदर यू आर गेटिंग इथाइल अल्कोहल और इथेनॉल, ठीक है, और ह्यूमन में या मैमल्स में बेटे वी आर गेटिंग लैक्टिक एसिड। इज दिस क्लियर बेटे? बट दिस इज व्हाट यू आर गेटिंग लिमिटेड अमाउंट ऑफ एटीपी फ्रॉम ग्लूकोज विदाउट एन इलेक्ट्रॉन ट्रांसपोर्ट चेन। गॉट इट बेटे? दिस इज द डेफिनेशन फॉर फर्मेंटेशन। इज दिस क्लियर?
ओके मैम।
ठीक है। अच्छा अब लेट्स टॉक अबाउट बेटे अल्कोहल फर्मेंटेशन। So let's understand it from the diagram. So ठीक है, ये आपका remember बेटा this is how it started. सबसे पहले साइटोप्लाज्म के अंदर जो ग्लाइकोलिसिस हो रहा है, this step will remain the same in both the cases. Glucose, ठीक है, because ATP is required to बेटा first phosphorylate the glucose. Glucose 3-phosphate बन जाता है, ठीक है. So you get ATP, 2 ATP molecules are used there. ठीक है, उसके बाद it converts into 2 molecules of pyruvate. And in the same process, बेटा pyruvate बनता है, तो you actually have these both of them are 3 carbon compounds. Carbon dioxide is also removed at this process, 2 molecules, but this pyruvate is converted into 2 acetaldehyde. Acetal जहाँ पे acetyl लग जाए बेटा, it means 2 carbon है. Pyruvate होता है 3 carbon, ठीक है, और acetyl जहाँ होता है 2 carbon. तो यहाँ से क्योंकि ये 2 molecules थे, तो हर एक के अंदर से एक कार्बन डाइऑक्साइड रिमूव हुआ, it means 1 carbon has been removed from each. तो अब ये 3 कार्बन से 2 कार्बन कंपाउंड के अंदर कन्वर्ट हो गए, ठीक है। सो दिस इज एसिटल्डिहाइड। एसिटल्डिहाइड पे फर्दर बेटा हाउ डज इट कन्वर्ट इनटू इथेनॉल, बिकॉज रिमेंबर व्हेन ग्लूकोज इज कन्वर्टेड इनटू पायरुवेट, NAD जो इनिशियली ऑक्सीडाइज्ड फॉर्म में होता है बेटा, इट एक्ट्स एज अ कैरियर मॉलिक्यूल या कोएंजाइम, इट देन टेक्स अप हाइड्रोजन, इट बिकम्स रिड्यूस्ड इन द प्रोसेस, ठीक है। अब क्योंकि आपके पास 2 मॉलिक्यूल्स पायरुवेट के बन रहे हैं, तो लाइकवाइज बेटा हर एक के अंदर से हमें एक रिड्यूस NAD मिल रहा है। So you have 2 molecules of NADH, ठीक है, which are reduced form. This acetaldehyde actually बेटा collects the hydrogen from it. तो initially जब हम लोगों ने aerobic respiration की बात की थी, ये NADH कहीं भी यूज नहीं हो रहा था. It was finally going inside the mitochondria और oxidative phosphorylation के अंदर further it was releasing this hydrogen. But anaerobic respiration के अंदर क्या हो रहा है बेटा, glycolysis के फौरन बाद pyruvate converts into acetaldehyde. This acetaldehyde is actually better collecting this hydrogen, okay. So in the process it becomes reduced now because it has gained the hydrogen from this reduced NADH. It becomes reduced और इसकी reduced form जो है that is known as ethanol. और अगर तुम ethanol का formula recall करो बेटा, तो it is C2H5OH, ठीक है। तो still बेटा it is a 2 carbon compound, ठीक है। So pyruvate क्या था, 3 carbon compound आपके पास इसके 2 molecules हैं, so you get 1 carbon dioxide from each, so एक carbon remove हो गया, ठीक है. We get acetaldehyde. Acetaldehyde will become reduced by collecting these hydrogen. Again, बेटा 2 molecules है, तो one hydrogen will be collected by each, ठीक है. It will become reduced and we are getting 2 molecules of ethanol, ठीक है, which is a 2 carbon compound now, ठीक है. अब इसको तुम जरा relate करो link reaction से. So I'm taking you to that slide now. See बेटे. So वहां पे क्या हो रहा था, link reaction that was taking place in the mitochondrial matrix. Anaerobic respiration की बात कर रही हूँ. 2 carbon compound वहां पे बना था, ठीक है. And then this 2 carbon compound was going into the Krebs cycle. तो अभी हमने anaerobic के अंदर क्या पढ़ा है, वही पायरुवेट जो है वो कन्वर्ट हुआ है 2 कार्बन कंपाउंड में. that has all happened inside the cytoplasm only, ठीक है. माइटोकॉन्ड्रिया के अंदर मूव नहीं किया, मगर वही पायरुवेट 3 कार्बन कंपाउंड कन्वर्ट हुआ है 2 कार्बन के अंदर, because this 2 carbon compound is only what can go into the Krebs cycle, ठीक है. Are you getting it बेटे? अगर तुम कंपेयर करो एरोबिक को, जी बेटे, जी क्लियर हुआ कि नहीं डिफरेंस दोनों का? ठीक है. सो एल्कोहलिक फर्मेंटेशन क्या चीज है बेटे? इट मोस्टली हैपेंस इन ईस्ट. इट कैन हैपन इन सम प्लांट्स आल्सो. सो बेसिक एम क्या बेटे? दिस हाइड्रोजन फ्रॉम द रिड्यूस NAD हैज टू बी पास्ड. अब अगर वो कहाँ पे पास किया बेटे? इथेनॉल या एसिटल्डिहाइड, एक ही चीज है, ठीक है. सो दिस इज नोन एज इथेनॉल आल्सो. इथेनॉल या एसिटल्डिहाइड, ठीक है. इट कलेक्ट्स दोज हाइड्रोजन एंड बिकम्स रिड्यूस्ड इन द प्रोसेस. जब ये रिड्यूस हो जाएगा बेटा, इट विल कन्वर्ट इनटू इथेनॉल. सो दिस इज द रिएक्शन आल्सो, ठीक है. पायरुविक एसिड जब एसिटल्डिहाइड में कन्वर्ट हुआ, उस वक्त कार्बन डाइऑक्साइड रिलीज हुई है, ठीक है. बट देन वंस दिस एसिटल्डिहाइड बेटे एक्सेप्ट्स हाइड्रोजन फ्रॉम दिस रिड्यूस्ड NADH, it becomes ethanol. You get the NAD in the oxidized form again, and then better this NAD can again be used in the process of glycolysis. So glycolysis का जो process है बेटा, it will keep on continuing in the cytoplasm, ठीक है. And in the end you will get आपको ATP के molecules यहाँ पे definitely 2 molecules मिल जाएंगे, ठीक है. But this NAD, ठीक है, it keeps on recycling. So once जैसे ही यहाँ पे बेटा हाइड्रोजन इससे रिमूव हुआ, ठीक है, it becomes oxidized, so it will be reused. So this process will keep on continuing, ठीक है. इन केस ऑफ एल्कोहल फर्मेंटेशन जहां पे इथेनॉल बन गया. अंडरस्टूड बेटे?
यस मैम.
ठीक है. सो दिस इज नोन एज एल्कोहलिक फर्मेंटेशन. अब लेट्स सी बेटे व्हाट हैपेंस इन. अच्छा एक दफा ये डायग्राम भी देख लो. दिस इज फ्रॉम योर बुक. So just for a review. So see beta, started from glucose, converted to pyruvate. This process beta requires ATP. ठीक है. तो शुरू में ATP यूज होता है because glucose has to be phosphorylated, but then at the end beta 2 ATP molecules are produced, and here beta they have shown you the entire cycle. So whichever diagram beta suits you, उसको आप ठीक है draw करके and then remember it. Pyruvate to ethanol, ठीक है. सिर्फ इस स्टेप के अंदर कार्बन डाइऑक्साइड रिमूव हुआ है, ठीक है. When ethanol accepts these 2 hydrogen and becomes reduced in the process, ठीक है. So anaerobic respiration जब हम alcoholic fermentation की बात करते हैं, आपके पास 2 end products आते हैं. One is ethanol and one is carbon dioxide. ठीक है. So 2 end products हैं, ethanol and carbon dioxide. Okay, so let's talk about the lactic acid fermentation. So the conversion of pyruvate that was formed in glycolysis in the cytoplasm to lactate with no release of carbon dioxide is lactic acid fermentation. So here's glucose, right? It converts into 2 molecules of pyruvate. But in this case, pyruvate accepts the hydrogen from reduced NADH, right? Because in the same process, oxidized NAD, accepted hydrogen become reduced, and this hydrogen is now accepted by pyruvate. It itself becomes reduced and converts into 2 molecules of lactate, right? So they're the only end product that we get is lactate. There is no carbon dioxide produced. So organisms that mainly use aerobic respiration can occasionally find themselves in a situation where there is not enough oxygen to continue on the aerobic pathway. So mostly this happens during exercise, right? You're pushing your body beyond the normal exercise routine. So in this situation, the respiratory pathway converts to anaerobic, and due to lack of oxygen after glycolysis is completed, the pyruvate molecules are converted into lactic acid molecules. And see, there are also 3 carbon molecules there. Just like pyruvate, as pyruvate is also 3 carbon sugar, right? So they convert into lactic acid, right? So this lactic acid can undergo two processes because it's an acid. If it gets deposited in the muscles, it would actually create the acidic environment there. It will lead to denaturation of the enzymes. It will lead to cramping. So this lactate has to be removed from the muscles also. So it can undergo two pathways. It can either be oxidized back to pyruvate, which is then channeled into the Krebs cycle for ATP production. So how this happens is basically because if the person is exercising more, he would be breathing deeply. He would be taking in more oxygen. So this actually, for this, the person needs extra oxygen actually. So this extra oxidation is referred to as oxygen depth, right? And this extra oxygen will then be used to convert this lactate into pyruvate, and this will all happen in the liver. Otherwise, the lactate can also be converted to glycogen for storage in the liver as well, right? But this lactate can be metabolized in two different ways. Either can be oxidized back to pyruvate. For this, we need additional oxygen, which is coming from the extra oxygen that the person is breathing in during exercise, right? Or it can be converted into glycogen. So that's the pathway. See? So the same glucose converts into pyruvate, but pyruvate itself gets reduced and converts into two molecules of lactate. They've shown you both the processes. Okay? This pyruvic acid can enter two pathways. It can either enter this or this. If it's clear, beta, मुझे बता देना.
Yes, ma'am. It's clear now.
Okay, beta. ठीक है. So in like anaerobic respiration में बेटे, we only get 2 ATPs generally. ठीक है. Aerobic में, because of oxidative phosphorylation, oxygen acts as a final electron acceptor, so we get around more than 30. ठीक है. But this is a difference. Okay then. ठीक है. Now let's talk about the respiratory substrates. Again, बेटे ऐसे type question में 6 marks का question इसका आ जाता है. So ठीक है. हमने जब बात की बेटे aerobic respiration की, we have generally बेटे discussed about glucose. बट ग्लूकोज के अलावा ठीक है अच्छा एसेंशियली ग्लूकोज जरूरी कहाँ पर है सो इट इज रिक्वायर्ड इन द ब्रेन रेड ब्लड सेल्स लिम्फोसाइट्स ठीक है बट अदर देन ग्लूकोज बेटे सेल्स कैन आल्सो ऑक्सीडाइज लिपिड्स ठीक है दे कैन आल्सो ऑक्सीडाइज अमीनो एसिड्स ठीक है सो दे कैन आल्सो बी यूज्ड एज रेस्पिरेटरी सबस्ट्रेट्स ठीक है जब जरूरत होगी तो एनर्जी कैन बी एक्चुअली एक्सट्रैक्टेड फ्रॉम दीज टू आल्सो अच्छा अब लिपिड्स को बेटे व्हेन एवर लिपिड्स आर रेस्पायर्ड कार्बन एटम्स आर रिमूव्ड इन पेयर्स जब हम ग्लाइकोलिसिस की बात कर रहे थे बेटा तो शुरू में 6 कार्बन से ग्लूकोज कन्वर्ट हुआ था 3 कार्बन में बट उसके बाद जितनी भी स्टेजेस थी बेटा इफ यू रिमेंबर द लिंक रिएक्शन वहां पे वो कन्वर्ट हो गया था 2 कार्बन में देन दिस वाज बीइंग टेकन इज द क्रेब्स साइकिल ठीक है और फर्दर उसमें कार्बन 1 बाय 1 करके रिमूव हो रही है बट जब लिपिड्स की रेस्पिरेशन होती है बेटे तो दिस कार्बन एटम इज बीइंग रिमूव्ड इन पेयर्स सो 2 कार्बन एट अ टाइम रिमूव होते हैं लिपिड्स के अंदर ठीक है and they are released as acetyl coenzyme A ठीक है and then acetyl coenzyme A can go into the Krebs cycle because Krebs cycle में बेटा this 2 carbon acetyl coenzyme A was entering ठीक है so it can then enter from the fatty acids it can enter into the Krebs cycle ठीक है because का reason क्या है lipids के अंदर बेटा they have a large molecular mass and they have more carbon hydrogen bonds there ठीक है तो जितनी हाइड्रोजन ज्यादा होगी बेटे फाइनली वही हाइड्रोजन आपका ऑक्सीडाइज NAD जो है वो एक्सेप्ट करके रिड्यूस होगा द सेम हाइड्रोजन विल गो इनटू ऑक्सीडेटिव फॉस्फोराइलेशन ठीक है तो जितनी हाइड्रोजन होगी उतनी ही ज्यादा फिर ATP प्रोडक्शन होगी दैट्स द रीजन लिपिड एक्चुअली गिव्स यू मोर एनर्जी एज कंपेयर टू कार्बोहाइड्रेट आर यू गेटिंग इट बेटे
यस मैम
सो अमीनो एसिड्स की भी जब हम बात करते हैं तो बेटा कार्बन हाइड्रोजन स्केलेटन्स the same these bonds are converted into pyruvate first ठीक है or they can convert into acetyl CoA so they can undergo 2 processes या तो वो pyruvate में convert हो जाएंगे then pyruvate beta will go into the same will convert into 2 like 2 carbon compound which is acetyl CoA then actually beta can go into the Krebs cycle ठीक है so we can get energy from both of them okay then so here beta these are the values so you can compare for yourself beta from carbohydrate we get 15.8 kilojoules per gram of energy और लिपिड में अराउंड डबल ठीक है उसका रीजन ये है बेटा इसका मॉलिक्यूलर मास ऐसा होता है इट हैज मोर नंबर ऑफ कार्बन हाइड्रोजन बॉन्ड्स ठीक है और जितना हाइड्रोजन है दैट हाइड्रोजन कैन गो इन द रिड्यूस फॉर्म इन द ऑक्सीडेटिव फॉस्फोराइलेशन मोर ATP विल बी प्रोड्यूस्ड ठीक है और प्रोटीन से कंपैरेटिवली अ लिटिल मोर देन कार्बोहाइड्रेट। ठीक है, सबसे ज्यादा एनर्जी मिल रही है लिपिड से। इज दिस क्लियर बेटे? ये टर्म फिर आपको ये वैल्यूज ना याद रखनी होंगी।
मैम याद रखती हूँ।
हाँ, ये आ जाती है पेपर के अंदर, बट जब ये पढ़ोगी तो यू विल रिमेम्बर देम। याद रह जाएंगी जब पढ़ोगी तो। ठीक है, अच्छा अब लेट्स टॉक अबाउट बेटा, क्योंकि रीजनिंग अगेन वही आपसे पूछ सकते हैं कि व्हाई वी आर गेटिंग मोर एनर्जी फ्रॉम लिपिड्स। ठीक है, लिपिड्स आर गिविंग अस दिस मच एनर्जी बेटे। नेक्स्ट उसके बाद नंबर था प्रोटीन का। ठीक है, और इन तीनों के मुकाबले में लीस्ट अमाउंट ऑफ एनर्जी वी आर गेटिंग फ्रॉम कार्बोहाइड्रेट्स। डिफरेंस इज बिकॉज ऑफ दिस रीजन बेटे। So difference in energy value can be explained by their molecular composition. ठीक है, specifically how many hydrogen atoms become available when the substrate molecules are broken down. So this is the reason बेटे। ठीक है, because lipid की जो molecular composition है बेटे, it has more carbon hydrogen bonds, it has more hydrogens there. ठीक है, so this is what will actually be available for making more ATP. This is the reason बेटे। Is this clear बेटे?
Yes ma'am.
Okay then. ठीक है, अगेन अब क्योंकि वही 6 मार्क का क्वेश्चन है बेटे, तो योर आंसर विल एक्चुअली बेटे बी रिटन इन दिस वे। ठीक है, सो ड्यूरिंग रेस्पिरेशन वी नो हाइड्रोजन एटम्स प्ले अ वाइटल रोल। ठीक है, सो सबस्ट्रेट मॉलिक्यूल्स आर ब्रोकन डाउन, हाइड्रोजन एटम्स बिकम अवेलेबल। ठीक है, सो दिस इज ऑल द सीक्वेंस ऑफ इवेंट्स बेटे दैट वी हैव ऑलरेडी डिस्कस्ड, सो जस्ट रीड देम। ठीक है, hydrogen carrier molecules called NAD and FAD, remember they were there in the Krebs cycle also. They pick them up, they become reduced in the process itself. ठीक है, and then these reduced NAD, reduced FAD, ठीक है, then transfer the hydrogen to the inner mitochondrial membrane. ठीक है, inner mitochondrial membrane के अंदर जाके बेटा they were releasing the hydrogen. So both reduced NAD and FAD, ठीक है, they release the hydrogen. Hydrogen then beta splits into hydrogen, electron and proton. Remember hydrogen has atomic number of 1, so वो split हो रहा था electron और proton के अंदर। ठीक है, so you get hydrogen ions also. So it gets splits up into these. The protons are pumped across the inner mitochondrial membrane to the intermembrane space. क्योंकि generally what was happening beta, these electrons were being accepted by the electron transport chain. ठीक है, because of the energy release, hydrogen was being pumped out of the inner mitochondrial membrane, it was going into the intermembrane space. ठीक है, and there was a proton gradient was actually being formed there. ठीक है, then because concentration of protons was more in the intermembrane space, they were coming back inside the inner mitochondrial membrane because of chemiosmosis beta, and this is how ATP was being produced. Are you recalling all these events beta? हमने aerobic respiration में पढ़ा था oxidative phosphorylation के अंदर।
Yes.
ठीक है, as these protons they flow back into the matrix, ठीक है, of the mitochondria, ठीक है, now they would like via ATP synthase they are then oxidized to form water. So the whole step beta we have studied, but जब तुम आंसर लिखोगी, so this is how beta you will talk about के hydrogen carrier molecules का। ठीक है, so अब in this case when we are talking about lipids mostly beta, when we have higher hydrogen content, it will result in greater proton gradient, ठीक है, across the mitochondrial membrane, and this will lead to more ATP formation via chemiosmosis. So does it make sense now beta? Okay, how are we getting more energy from lipids as compared to carbohydrates?
Yes ma'am.
Okay. So fatty acids का क्या reason है बेटा? They are made up of long hydrocarbon chains with a lot of hydrogen atoms. ठीक है, and these hydrogen atoms are released when the lipid is broken down. ठीक है, so this is the reason. If they ask you about how lipid is able to generate more ATP, so that's the reason बेटे। एक दफा इसको go through भी कर लो, then we'll move on to the next one. As far as amino acids are concerned, since they are proteins, they only respire aerobically when all other substrates have been utilized. The main reason for that is actually because they have these essential functions elsewhere in the cell. right? And when we talk about essential functions, you know that amino acids, they are required for making proteins, right? And proteins which have their structural requirement, for example, they're required in the cytoskeleton, and they have functional requirements, for example, they have their enzymatic roles also, right? So these will be utilized as a last resort. ठीक है? Then same as I was saying कि बेटा you can be asked, ठीक है? पेपर में क्वेश्चन आ सकता है. Why different respiratory substrates have different energy values? ठीक है? So एक इन्होंने example question दिया है. Explain why carbohydrates, lipids, and proteins have different relative energy values as substrate in respiration aerobic conditions. So you will talk in terms of these. तो आपकी explanation यही होगी, ठीक है? But then you can talk about lipids here in detail also. ठीक है बेटे? Now let's move on to respiratory quotient.
Yes, ma'am.
ठीक है? अब respiratory quotient क्या है? It is simply the ratio of carbon dioxide molecules produced to oxygen molecules taken in during respiration. So ratio है, it doesn't have any unit. So this is how beta carbon dioxide will be in the numerator, oxygen will be in the denominator. तो इसके लिए जनरली पेपर में ये भी हो सकता है कि आपको एक केमिकल इक्वेशन दे जो पहले तुम्हें बैलेंस करनी हो, और बैलेंस करने के बाद you have to find the respiratory quotient. ठीक है? So here they have given us example for glucose also. ठीक है? कि ग्लूकोज का बेटा वी नो के ग्लूकोज मॉलिक्यूल इज C6H12O6, और एक बैलेंस्ड इक्वेशन के अंदर यू हैव six ऑक्सीजन एटम्स आर कंज्यूम्ड, 6 कार्बन डाइऑक्साइड आर प्रोड्यूस्ड. तो अगर उसको उसी फॉर्मूले के अंदर बेटा अगर हम रखते हैं, ठीक है, बिकॉज इस रेशियो ऑफ कार्बन डाइऑक्साइड प्रोड्यूस्ड टू द रेशियो ऑफ ऑक्सीजन कंज्यूम्ड, सो 6 डिवाइडेड बाय 6, सो ग्लूकोज का जब हम रेस्पिरेटरी क्वोशिएंट निकालते हैं इट इज इक्वल्स टू 1. ठीक है? But all three of them have different respiratory quotients: carbohydrate, lipid, and protein. ठीक है? और उसका रीजन बेटे सिंपली क्या है कि बिकॉज द नंबर ऑफ कार्बन हाइड्रोजन बॉन्ड्स आर डिफरेंट, सबका मॉलिक्यूलर फॉर्मूला डिफरेंट होगा. ठीक है? सो दे विल बी बैलेंस्ड आल्सो अकॉर्डिंगली. ठीक है? सो इफ देयर आर मोर कार्बन हाइड्रोजन बॉन्ड्स, it means more hydrogen atoms can be used to create a proton gradient. More hydrogen means more ATP molecules can be produced. ठीक है? और अगर बेटा ज्यादा ATP हमें बनाना है तो इन फैक्ट बेटा वी नीड मोर ऑक्सीजन ऑल्सो. ठीक है? सो मोर ऑक्सीजन इज देयरफॉर रिक्वायर्ड टू ब्रेक डाउन द मॉलिक्यूल, क्योंकि डेफिनेटली इससे वाटर में भी तो कन्वर्ट होना है ना बेटे. ठीक है? व्हेन ग्लूकोज इज एरोबिकली रेस्पायर्ड, सो वी जस्ट वर्कड इट आउट बेटा रेस्पिरेटरी क्वोशिएंट ग्लूकोज के केस में तो बनाएगा. सो लेट्स, ठीक है, दे हैव गिवन यू एग्जांपल्स, दीस डेफिनेटली बेटा यू हैव टू रिमेंबर. ठीक है? ये आपको पेपर में कोई ना कोई सिनेरियो होगा, कोई ना कोई केमिकल इक्वेशन होगी जिसके थ्रू यू विल बी एक्चुअली बेटा यू आर सपोज टू कैलकुलेट इट. सो आंसर आपका नियरली, ठीक है, इन वैल्यूज के आसपास आना चाहिए. ठीक है बेटे? सो अगेन बेटे सम क्वेश्चंस में आस्क यू टू सजेस्ट which substrate is being respired. So first you will be asked to find out the respiratory quotient. Based on that, they will ask you which substrate was that. Was it carbohydrate? Was it lipid? Was it protein? In जो आपका जो आंसर होगा उसके हिसाब से बेटे you will have to tell. Is this clear बेटे? जी बेटे?
Yes, ma'am.
Okay. So here is the same better. They have written the formula. We have already discussed that, but this is the formula. Okay, moles या molecules of carbon dioxide given out divided by moles or molecules of oxygen taken in. So we have already discussed the example of glucose. Now this is an example where they have given for lipids, और ये अक्सर बेटा पेपर के अंदर भी आ जाता है. ठीक है? So this is the molecular formula. What they have given you here बेटा is an unbalanced chemical equation. ठीक है? So इसको तो पहले आपको बैलेंस करना पड़ेगा. सबसे पहला काम बैलेंसिंग है. बैलेंस करने के बाद बेटा you will look at the concentration of oxygen and the concentration of carbon dioxide. ठीक है? Then this is the formula you would be using. ठीक है? अब इस केस में because there are 25 oxygen, ठीक है, and 18 carbon dioxide, so the value is coming as 0.72, जो round off करके बेटा 0.7 आ रहा है. And now if they ask you कि what is the substrate, then you can tell कि this is lipid. ठीक है बेटे?
Yes, ma'am.
ओके देन. ठीक है? तो ये तो हम रेस्पिरेटरी क्वोशिएंट बेटा डिस्कस कर रहे थे एरोबिक रेस्पिरेशन के लिए. सो लेट्स सी बेटा हाउ कैन वी कैलकुलेट रेस्पिरेटरी क्वोशिएंट्स फॉर एनाएरोबिक रेस्पिरेशन. ठीक है? एनाएरोबिक रेस्पिरेशन, नो यू नो बेटा देयर इज नो ऑक्सीजन इन्वॉल्व देयर. तो अगर हमें रेस्पिरेटरी क्वोशिएंट निकालना भी है बेटा वी नीड ऑक्सीजन. ठीक है? अब अगर वो अवेलेबल ही नहीं है तो रिमेंबर इन केस ऑफ लैक्टेट फर्मेंटेशन इवन कार्बन डाइऑक्साइड वाज नॉट प्रोड्यूस्ड. इफ देयर इज नो ऑक्सीजन अवेलेबल एंड नो कार्बन डाइऑक्साइड प्रोड्यूस्ड आल्सो, सो आप यहाँ पे रेस्पिरेटरी क्वोशिएंट इसका तो किसी भी सूरत हम निकाल ही नहीं सकते. because both of them are not there. ठीक है? So for these beta it is not calculated at all. ठीक है? But जब हम alcohol उसकी alcohol fermentation की बात करते हैं, the thing is oxygen is not there but carbon dioxide is still produced. ये product तो बन रहा है आपका. तो इसके केस में बेटा you get an answer as an infinity, because if अगर हम उसकी यही chemical equation use करें, the same glucose बेटे, because there is no oxygen this time, it gets broken into ethanol and carbon dioxide. अब formula तो हमारा यही है, ठीक है? तो carbon dioxide अगर if after balancing the equation we are getting 2 molecules of carbon dioxide, there is no oxygen, ठीक है? So you get infinity. So in some question बेटा जो values हमें मिलेंगी अक्सर वो इस तरह की होंगी, मतलब more than one generally कोई ना कोई value होगी. So उन values के through you'll get to know कि we are talking about alcohol fermentation. ठीक है? क्योंकि ग्लूकोज की 1 है, ठीक है? उससे ज्यादा कोई भी, क्योंकि इन्फिनिटी है, बट पेपर में जब क्वेश्चन देखो तो वैल्यूज इस तरह की 7 पॉइंट समथिंग 8 पॉइंट समथिंग, तो कोई वैल्यू जो बहुत ज्यादा मोर देन 1 आपको लगे, इट मींस वहां पे अल्कोहल फर्मेंटेशन हुआ है. ठीक है बेटे?
ओके.
सो दैट्स द डिफरेंस बिटवीन रेस्पिरेटरी क्वेश्चंस फॉर एरोबिक एंड एनारोबिक. ठीक है? और द सेम इट एज वी जस्ट डिस्कस्ड. आपको मेक श्योर करना है कि इक्वेशन फुली बैलेंस्ड हो. ठीक है? तो ये 2 या 3 नंबर का सवाल आता है. So you will actually have to work this out. Is this part clear, Bete?
Yes, ma'am.
So in these cases, there might be a possibility that the organism is respiring both aerobically or anaerobically, right? So the values that we can get maybe of 9 or 10. So if it's not 0, but maybe it's 0.2, maybe 0.1, you can get a value which is definitely more than 1. So it tells you that the organism is respiring both aerobically or anaerobically. So sometimes when the RQ value changes, the respiratory quotient changes. Like as we know, for glucose it's 1, for lipids it's 0.7, for proteins it's 0.9. So if the RQ value changes, it means that the substrate being respired has also changed. Now, some cells might also be using a mixture of substrates in respiration, right? So you can get sometimes a respiratory quotient value of 0.85. which is actually like, if you think about it, it means that the organism is respiring using both carbohydrates and lipids as a source, because the RQ value for glucose is 1 and the RQ value for lipid is 0.7. So it's somewhere in between, right? So it means that the organism is respiring using both the substrates. So under normal cell conditions, generally this is the order that these substrates are used. So first carbohydrates, then lipids, and finally proteins. As we just discussed, the proteins are actually required in other cellular functions, so that these are the last resort that actually an organism would go for, right? Then the respiratory quotient can also give you an indication of under or overfeeding as well. If the respiratory quotient value is more than 1, it suggests that excessive carbohydrate or caloric intake is being taking place. And if it is less than 0.7, so it could also suggest that underfeeding is also present there. All right then, so in order to investigate the respiratory quotient, an instrument known as Respirometer is used, and it is used to measure and investigate the rate of oxygen consumption during respiration in organisms, which can be seeds or invertebrates, or it can also be used to calculate the respiratory quotient, right? So for this, I want you to have a look at a video, and then we'll come back and talk about the rest of the things.
So we're going to be looking at the use of a respirometer today. We're going to go through all of the pieces of equipment here, the purpose of each, and then what we'll be measuring. And it's all to do with changes in volume and therefore changes in pressure caused by gases absorbed by whatever living organism is placed inside of this experimental tube. So let's have a look then at the equipment. You would typically have 2 test tubes. One is there as a control and the other is our experimental tube. And they are attached together by what we call a manometer. And this is this really, really thin capillary tube which has coloured liquid inside of it. And against that, there'll be a piece of plastic typically, which has a scale on it, like a ruler, so you can measure how far this coloured liquid moves over a period of time. Now, the top of each test tube has to be completely airtight because we're going to be looking at changes in gas volume and pressure So therefore we have to make sure no gases are entering or leaving, so completely airtight with a bung. We do still have, though, coming out of the bung a capillary tube, which would have a 3-way tap, and that's what this label would be, 3-way tap. And on the control tube we also have a syringe. Now the point of this is you will need to, if you want to do repeats, reset your equipment so you can move the coloured liquid back to the start point, whereby both points are at exactly the same level. And you can do this by either pulling up or pushing down on the syringe to change the volume of gas inside and therefore move the liquid back to the start point. Our experimental tube will have a living organism within it that will respire. Now I'm using maggots as the example today, but just as likely you could do this experiment with woodlice, or sometimes with plants, in particular peas is a common example, fresh peas which should still be respiring. In both test tubes we have soda lime. The purpose of this powder or the granules of soda lime is to absorb any carbon dioxide. So then we'll know any changes in volume are just to do with oxygen being absorbed. For this bit here as well, you'll have a piece of gauze or a metal mesh just to make sure those maggots can't touch the soda lime, because it would be harmful to their skin. It could be an irritant, so we need to make sure they don't touch that soda lime. Our control tube, exactly the same, except if we want to prove that it's definitely the organism that is respiring and nothing else causing the changes, then we need to put an inert object. So we've got glass beads here as an example, because they won't be respiring, but you'd put the same mass of glass beads in as maggots just to keep everything consistent. So what we'd then expect is, if we have this all set up, then close the taps to make sure it's completely airtight. You then set a timer and look at how far the coloured liquid moves over a set period of time. And what we'd expect to happen is the maggots will be respiring, and for aerobic respiration they'll be using up oxygen, and that will then mean, to replace the oxygen used, oxygen from the gas, the air within the experimental tube, will diffuse into the maggots. The maggots will also be producing an equal amount of carbon dioxide as they respire, but that carbon dioxide is going to be absorbed straight into the soda lime. So the effect that we'll see is the volume of gas inside of the experimental tube will decrease, because the oxygen is moving into the maggots. The carbon dioxide that's produced is absorbed straight away out of the local atmosphere in that tube into the soda lime. So if the volume of gas decreases, that will mean the pressure will drop inside of our experimental tube. Now that won't be happening in the control tube because the glass beads aren't taking in any oxygen. So comparatively, the glass beads, the control tube here, will have a higher pressure compared to the experimental tube. And because the pressure is comparatively higher in the control tube, that will then force the air around and the liquid inside of the manometer tube, and therefore the liquid will start to move towards the experimental tube. So if we left it for 5 minutes, we might expect to see the liquid has moved round. So at the end of that 5 minutes, what we could then do is work out what was the rate of respiration. And the way to work this out is, it's the volume of oxygen absorbed over a particular period of time for a particular mass of maggots. Now the volume of oxygen, the way we can work that out is the volume of the cylinder for which the oxygen was moving through. So we need to know what is the area of the cross-section of the manometer or this capillary tube, and the length is the distance that it travelled. So if we imagine each of these lines was representing 5 millimetres, for us it might have moved, if that was the start point, 5, 10, 15 millimetres. So once you've worked out the volume, then you are dividing that by the period of time you left it for. So we said in our example 5 minutes, and we also divide it by the mass. And the reason you have to take into account the mass is so it is a fair comparison. Because if you want to work out the rate of respiration for maggots, you'll end up with a much higher value if you have more maggots present. But that doesn't mean those maggots were respiring faster, it just means there were more of them. So that's why we always have to take into account the mass of the maggots, so it is a fair comparison. So for that reason, the unit is always a 3-component unit. It's always a unit for volume, per unit time, per unit mass. So for example, it could be centimetres cubed per minute per gram. So we're going to have a go then at some questions. So at this stage you might want to pause, get your pen out and your paper and have a go at these exam style questions. Just to give you an overview of how I've split up the types of questions, the top 2 are linked to the apparatus setup. So why does it have to be airtight? What is the purpose of the soda lime? Then we've got application of your knowledge. So explain why the liquid moves to the left and try and include as many key terms as possible. Then we've got our maths over on this side. So can you remember what the units would be? 3 marks as a reminder, there's always 3 components to those units. And then actually calculating the rate of reaction. And again, you have to include the units in your answer. So pause the video, have a go at these. It should take you, I'd say, about 5 to 10 minutes to answer these questions. So number 1, why does it have to be airtight? Well, the whole reason the liquid moves is because of pressure changes. And if any gases could enter or exit, then it would mean that the pressure changes are no longer due to the respiring maggots, it's due to another factor. So it's going to affect the movement of the liquid. So that's why we need to make sure it's completely airtight. The purpose of the soda lime is to absorb the carbon dioxide produced. And that's because the volume of oxygen that will be absorbed by the maggots will be equal to the volume of carbon dioxide produced in respiration. So if that carbon dioxide was not absorbed, the volume would always remain constant, the pressure would remain constant, and that liquid would not move, and therefore we'd have no way to measure the rate. So it absorbs the carbon dioxide, so we are able to actually measure the volume of oxygen absorbed by the maggots, and we use the volume of oxygen absorbed to represent the rate of respiration. The next one was explain why the liquid would move to the left. Now sometimes they'll add in an extra question and they'll actually get you to work out which direction would the liquid move first. And it's not always set up, the experimental tube's on the left, sometimes it's on the right, but the liquid will always move towards the experimental tube. And the reason for that is the maggots are respiring, so they're going to be having more oxygen diffuse across their surface for more aerobic respiration. Carbon dioxide which is produced will be absorbed by the soda lime, so we'll end up with a decrease in volume in the experimental tube. That will cause a decrease in pressure. Comparatively, the control tube has a higher pressure now compared to this lower pressure in the experimental tube. Therefore, it's going to force that liquid, that coloured liquid, around towards the experimental tube. So the units, 3 parts, is always a unit for volume, per unit time, per unit mass. So I've used this as an example, but it could equally be millimetres cubed. Most likely it is per minute for this. So if it was per second, seconds isn't really long enough to see a change. And per grams is the most common because whatever you do put in the experimental tube has to be small enough to fit in a tube. So you're going to be measuring it in grams, so per gram. The last one we had was the rate. So using that information and the formula, the rate of reaction is the volume divided by time times mass. The volume, pi r squared times length, and our radius is half the diameter of the manometer tube. So that is 1.5. So pi 1.5 squared. The length is the distance the coloured liquid moved, and the coloured liquid moved 5 centimetres. So that then gives us our volume of oxygen absorbed, but to have it as a rate, we need to divide that by the time it took to absorb that amount, that volume, and what mass of maggots absorbed that amount. So it was 5 minutes and 5 grams. That's why I've done it, divided by 5 times 5. And that works out to be 1.41 centimetres cubed per minute per gram. So that is our respirometers, one of the ways to measure the rate of respiration. So just some of the key points to remember. The respiring maggots, they are absorbing, they're taking in that oxygen for respiration. The carbon dioxide which is produced is equal to the volume of oxygen taken in, but it's absorbed by the soda line. That then causes a decrease in volume of the gases and therefore a decrease in pressure of the gases in the experimental tube. Because we have that lower pressure in the experimental tube compared to the control tube, that forces the coloured liquid to move towards the experimental tube. Last key point I put down was the units, because this is quite a common either 2 or 3 mark question to know the units for rate of respiration. So centimetre cubed or whichever unit of volume. The idea is it has to be cubed, per unit time, per unit mass.
So in the video, we saw that how this respirometer can be used to measure the rate of oxygen consumption. Now we are talking about how the same apparatus can be used to measure the respiratory quotient. And as you know, the formula is carbon dioxide produced to oxygen consumed. So that's how it works out the ratio. Okay then. So for this purpose, since we have already set up the experiment, we have taken, let's say, 2-3 readings, and because we were calculating the rate of oxygen consumption, so we have actually got the value for oxygen only from the first experiment, right? Let's say you have got this value as 6. But how do we get the value for carbon dioxide now? For this, what we need to do is we need to eliminate soda lime here from the experimental setup as well as from the control, so that no carbon dioxide is being absorbed by the soda lime now, right? So how are we actually going to measure that? Now, in this case, if for instance, the same amount of carbon dioxide was being produced, let's say it was 6. So what would happen? You would have the same value here. The manometer would not move at all, right? Because if the same amount of oxygen is being consumed and the same amount of carbon dioxide is being produced, so this manometer will stay at the same position, right? So in this case, we know, yes, the respiratory quotient is 1. But every time this will not be the case, right? Sometimes more carbon dioxide will be produced as compared to oxygen that is consumed, and then vice versa will also be true. So in that cases, what we need to do is take this value of oxygen that you have already taken as X, right? This value will remain same. In cases when carbon dioxide concentration is more, right? So in that case, you would definitely see a value that is increased in this case. So what will you do in that case? The original value that we had, and we are going to add the new value to it, right? Because now the value has increased. This is what will tell you the respiratory quotient in the case when more carbon dioxide has been produced. Alternatively, if less carbon dioxide has been produced as compared to oxygen that was consumed, the formula would be X minus Y upon X, right? So this is how we are going to calculate the respiratory quotient. So in terms of more carbon dioxide produced, in fact, X plus Y upon X, and in terms of less carbon dioxide produced, we are going to go for this formula, right? So this is how respiratory quotient is calculated when we want to know how much carbon dioxide has been produced. So in the video, you also must have noticed that the unit that they were using were a centimeter cube per minute per gram. So in your book, the unit that they have used, they're not taking mass into account. So they're simply using cubic centimeter per minute. So you may stick to the same, right? But right now, in this particular part, we're talking about how temperature would actually be affected and how would we see this effective temperature using a respirometer. So here is the procedure. So first and foremost, we are going to measure the oxygen consumption. So that is the first thing that we do, right? So we have this setup. So the first thing that we do is measure the oxygen consumption, right? Okay then, so you set up the respirometer, you run the experiment with both the tubes in controlled temperature water bath, because right now we are talking about the effective temperature, right? So both the test tubes are in controlled temperature water bath. You're going to use the manometer reading to calculate the change in gas volume within a given time. So step number 1 remains same. Then you're going to reset the apparatus, allow air to re-enter the tubes via the screw cap, and reset the manometer fluid using the syringe. And then you're going to change the temperature of the water bath and allow the tubes to acclimate. Then close the screw clip to begin the temperature, because we reset the experiment and we want to take a new reading now. Now you run the experiment again. Use the manometer reading to calculate the change in gas volume in a given time. And now this change we are calculating as Y cubic centimeter per minute. And then you repeat the experiment several times at different temperatures. And this is what you're looking at, okay, how different temperatures affect the rate of respiration. So just to recap, the volume of the oxygen consumed will be used or found out using this formula, right? So you will work out the diameter of the capillary tube, you will work out the radius, and the distance that is moved by the manometer is taken as h, and the value for pi, 3.142, right? And we are calculating that per minute. So you will work out the volume of the gas or oxygen that is consumed, right? So the rate of oxygen consumption is often taken as the rate of respiration for the organisms. And then different volumes of oxygen consumed obtained for different temperatures can be presented in a table or a graph formed to show the effects of temperature, right? So this is how we can show the effect of temperature using a respirometer. So this is the graph drawn. So you can see, so this is the relative respiration rate against the temperature. And you can see as the rate of respiration is increasing as the temperature is increasing, right? But after 40, the rate of respiration decreases. And the reason being, the enzymes actually start denaturing at high temperatures, right? So this is how we are going to see the graph. And besides the respirometer, the effect of temperature and substrate concentration can also be investigated using a redox indicator, which can change color when it is reduced or oxidized. So the redox indicator that can be used is DC-PIP and methylene blue. So both of these can be used. And these actually are used to investigate the effect of temperature and substrate concentration on the rate of aerobic respiration in yeast, basically, right? So what these indicators do, these dyes generally they're added to a suspension that contains living yeast, and these do not damage the cells, right? So that's the mechanism actually. These indicators, they go from blue to colorless, right? Because in different stages of aerobic respiration, hydrogen is being removed from the substrate molecules, and then that hydrogen is transferred to the final stage of aerobic respiration, oxidative phosphorylation, via the hydrogen carriers, which are NAD and FAD, right? Now, whenever we are using these indicators, these DC-PIP and methylene blue, they will actually act as those hydrogen acceptors. They will take up the hydrogen, and in that case, they will get reduced. So the faster the rate of respiration, the faster the rate of hydrogen release, and faster the dyes, they get reduced and change the color. So this is what we're looking for. The faster they go from blue to colorless is how fast the rate of respiration is taking place, right? So the rate of respiration is inversely proportional to the time taken, or 1/T, right? So this is the mechanism if we are calculating or using a redox indicator, right? So we can use the same dye for investigating the effect of temperature as well as investigating the effect of substrate concentration. So if we are working out the effect of temperature, so what we are going to do, we are going to add test tubes that contain the yeast suspension and to a temperature controlled water bath, right? And then you're going to record the time taken for the color change to occur once the dye is added. So once you put it in the temperature controlled water bath, excuse me, and then you're going to repeat the same procedure across a range of temperatures. So you can use different temperatures. You can use 30, 35, 40, and 45 degrees Celsius. And then you see that how long does it take for that dye to go colorless. Secondly, you can actually also see the effect of substrate concentration also. And in this case, you may add different concentrations of a substrate, right? Into the suspension of yeast cells, and then record the time for the color to change again from blue to colorless. And you can use different concentrations like 0.1% glucose, 0.5% glucose, 1% glucose, or you can use different substrates as well. You can use glucose, maltose, fructose, right? So you can use different substrates also to calculate the effect on the rate of respiration in yeast. So it is, however, imperative that you control other variables if you are investigating temperature or substrate concentration. So you have to keep some variables under control. So volume of the dye added. If there is more dye molecules present, then the time taken for the color to change will definitely be longer. So you have to make sure that the dye molecules that you're using are like same in all of them, right? Volume of yeast suspension. When more yeast cells are present, the rate of respiration will be inflated. Then type of substrate. Yeast cells will respire different substrates at different rates. Concentration of the substrate as well. If there's a limited supply of substrate in one tube, then the respiration of those yeast cells will be limited. And any increase or decrease in the temperature can affect the rate of respiration due to energy demands and the kinetic energy changes. The temperature of the dye being added also needs to be considered. So these are a few variables that we need to be taken care of, and they have to be actually kept in control. So here's just an exam tip. Although we know that DCP by methylene blue will go colorless, but the thing is the yeast suspension itself is slightly yellowish. It's a like cloudy solution, right? So it would have the same, it would have a slight color. So it's not that it would go colorless. So this means when the dye changes to colorless, there may still be an overall yellow color in the test tube. If this is the case, it can be useful to have a control tube containing the same yeast suspension, but with no dye added. Then you can tell whether the dye has completely changed color or not, right? So its usual color will be a little yellowish, brownish. All right. So the respirometer that we have actually seen here in the previous examples also is set up for measuring the rate of aerobic respiration. It cannot be used to measure the rate of anaerobic respiration as no oxygen is consumed, because here we are actually recording the rate of oxygen consumption, right? So again, if the temperature is more, just think about it. At higher temperatures, there are chances of proteins to become denatured, and then they will become unable to carry out the function. At lower temperatures, molecules and enzymes don't collide very frequently as they don't have a lot of energy, right? So the same trend can be seen in the rate of respiration, right? Because these reactions, they rely on enzymes, right? So this is a last part about the respirometers. And now in the next lesson, we'll talk about anaerobic adaptations of rice plants.